the pathway selection of commissural neurons. So these commissural neurons are there, and what they are doing, this is an interesting thing. So right here is your spinal cord, and these neurons are somewhere here, okay? This is the central hole there. And they actually move like this. They move like this, from here to here. Right here is your floor plate. And these are your commissural neurons. So they are growing, the neuron here, the axons are growing like this, okay? Now, right here on the floor plate, why they move like this? Right here on the floor plate, you have a lot of here, what is secreted in the floor plate? Sonic hedgehog. Right here is the sonic hedgehog. Actually, sonic hedgehog also act as attractant signal. So sonic hedgehog receptor are called patched. There are two types of patched. Patch 1 and patch 2. And that activate SMO, SMO, right, that activates SMO. And when the SMO is activated, you get further signaling there. Now, actually, there are not only sonic hedgehog in the body. This was sonic hedgehog in the body. There is also desert hedgehog. That desert hedgehog is there in our body system during the development. There are 3 different types of hedgehog signaling working. And desert hedgehog is required in bone development. Then there is also Indian hedgehog. That Indian hedgehog is in gonad development. So 3 different types of hedgehog signalings are there. Sonic hedgehog is mainly in nervous system, right? So we are talking about sonic nervous system. Just wanted to give you these names also so that someone somewhere, some exam, someone asks you, so you know that there are other 2 signals also. Desert hedgehog and Indian hedgehog. So now we are saying sonic hedgehog is there. So that means these are moving on this side, right?
Sorry. So patch 1 and patch 2 activate SMO.
Yeah.
What is SMO?
SMO is the downstream signaling molecule. So this is the receptor, right? So this is patch receptor. Once patch is activated, a protein called SMO is activated. And when SMO is activated, that causes the further changes in those objects, basically. Okay? So there's PBOK and all those pathways are there. But we need to only remember for this purpose, so this is only for our purpose. Now, sonic hedgehog is there, very high sonic hedgehog, we know that. So they are attracted towards sonic hedgehog, okay? But in the midline right here, in the midline, so this is the midline. Midline, you have lot of, I'm drawing 2 different colors so that we'll use the same color for all things. So lot of natrin or lot of natrin there, okay, and then also lot of slit there. Okay, so two things are happening. Natrin is a attractant, right? Slit is a repellent molecule. So what happens? These neurons, I'm just drawing two neurons, there are many of those, right? So these neurons, they actually don't stay like this. What they do, they actually at certain location, for example, they actually cross from this side to this side and cross from this side to this side. Okay, so this commissural neuron, so axons of commissural neuron, they cross midline. Okay. So then how they cross the midline? That's kind of very interesting question. So what is happening, let me draw it here. So I'm just drawing the midline. So your midline is here. Okay. So in the midline, you have a lot of slit there and lot of natrins are there. Right, so this is color coded. So what happens that you have a so here is your axon, and this is the growth cone of the axon. It's coming like this. Okay. So what is happening here? You have a lot of sonic hedgehog, right? And that sonic hedgehog is going there, right here. This is your patch, and that activates smoke. So it's coming here. Okay. Now you will say that why this axon does not go to our midline, right? Because you have natrin here, and these neurons also have, so this is the DCC right there. Now DCC is the receptor for natrin, right? So natrin should also go there and attract it. Making sense? But it does not happen. It doesn't happen because right here, so the full form of robo is roundabout. So roundabout abbreviation is robo. So remember that also. Okay. So they also have the receptors here for robo, for slit. So this receptor is there. This is, now robo have robo 1, robo 2, robo 3. This is robo 1 receptor is there. Okay. So what happens that the slit, so this is slit, come here, and slit is being, lot of slit here, so it's kind of like coming here, slit bind here to the robo 1. Okay? When slit bind to robo 1, what it does, robo 1 is now active, right? That receptor is activated. And that cause this actually come here and so robo 1 activation block DCC. So this is now blocked. So this, you said this is now blocked. So if this is blocked, say this is like this, blocked. So if this is blocked, net pin cannot attach to it. Okay? So, what slit is? Slit is chemorepellent. Slit is saying, don't come on this side, right? Slit is chemorepellent. But slit activation also block DCC, so this netlin cannot bind, so this netlin cannot either attract it or it. Making sense? So then there is only one signal active, that is sonic hedgehog signaling, and that is telling them to move down. So far is okay? Now the time is to move on this side. Say, now this is the time to move on this side. It has to move on this side. Then what happens when it needs to move? Okay, now we'll draw the same thing here. Lot of slit, lot of slit, lot of slit, lot of slit, lot of slit. And lot of netlin there. Lot of netlin. Now what happened at this stage? Now it needs to move on that side. So this is coming down, right here, say this is here, right here. Now this is the position where it's supposed to cross the midline, okay? Again, you had sonic hedgehog. Here you had active small, right? So what is going to happen? Sonic hedgehog level is still there. At that time, these axons, they start expressing robo 3. Okay. So here, robo 3 is there. Now, here is the interesting thing. So far, I'm strictly saying this is chemoattractant, this is repellent, this is this. These receptor, what type of receptor is there? That can change the behavior of a particular chemoattractant or chemorepellent. So, a new receptor, robo3, start expressing there. Okay. When this thing happens, what happens? This robo3 activation, what it does, robo3, let me write here, robo3 blocks robo1. So, robo3 activation, and right here is robo1 blocked. So it blocked. So far it is okay. Then what happens? Robo3 activation send signal to nucleus to make 14-3-3. This is the name of the protein. 1433 protein is made. That 1433 protein come here, so this is say this is 1433 protein, that come here and that block SMO. So block SMO. So far it is okay. So here three things are happening. Number one, Robo3 activation take place. Then second thing, Robo3 activation block Robo1. So if Robo1 is blocked, that's the one thing. And then Robo3 activation sends signal to nucleus to make 1433 protein, and 1433 protein block inactivates SMO. So two things have happened because of this. Number one, sonic hedgehog signaling is no more working. Making sense? And then now this Robo1 is blocked, so slit cannot bind to it. If slit cannot bind to it, then you have this receptor for natrin. Now this natrin receptor is open. Right? What Robo1 was doing? Robo1 was activating and blocking the DCC here. So this DCC was there, this DCC was blocked. Right? Now DCC is open. If the DCC is open, what is going to happen? This natrin is going to come here and bind here. Right? So if natrin is binding here, so now this axon know that hey, here is the attractant. Okay? So what is going to happen? This axon will now move on in this direction. No axon is moving in this direction. Making sense so far? The same thing is happening to the axon which is coming from this side also, because that has to move on this side. So far it's okay? Now it has reached here and say, okay, then what happened? So now say that this has come here. Okay. When it reaches here, then you want to go further away from this. You don't want to stay it at the midline. Okay. And then what happened? At this step, Robo3 downregulate. Robo3 downregulate. Okay. So again, if you have sonic hedgehog, so just draw it here. Sonic hedgehog was there. So this was the gradient of sonic hedgehog, right? Very low, very high, down. Making sense? Now what happened? Robo3 down regulates. So you in this area you have a lot of sonic hedgehog around there. But natrin is there. Now Robo3 is down regulated. Down. So therefore 1433 also down, right? That means SMO activation. So it can sense the SMO is here. But natrin is also there, right? So if Robo3 is down, Robo1 blockade will be gone, right? So the Robo1 here, so this is the Robo1. Robo1 will get slit. And when that is going to happen, Robo1 binding to slit, that is going to this is Robo1 active, and this is now DCC blocked, right? Now Robo1 is a chemorepellent signal. Natrium is attractive, but natrium is blocked. Sonic hedgehog is attractive, right? So what is going to happen? This is going to move further, and because now you have Robo1 active here, it will not come back. Making sense? So in this way, it's going to cross the midline. So let's go back and talk about these steps once more. So we are saying that these commissioner neuron, they actually go from left to right side of the spinal cord. Now, if there is mutation in any of this, that can be devastating. Even the slightest mutation in this can actually cause the bigger problem in the eye coordination. The two eyes, they cannot see the same thing at one time. It's not strabismus. Strabismus is the muscle problem, but this is more of a neuronal problem. So, commissioner neuron are there. Their axon cross the midline, but we are saying in the midline there is a lot of slit there, and there is a lot of natrium. Natrium is chemoattracting, but this slit is chemorepellent. So if there is chemorepellent there, then how it crosses the midline? We are saying the floor plate is having a lot of sonic hedgehog, and sonic hedgehog bound to patch activates SMO, and that causes attractive signal. Whereas slit come and bind to Robo1. When it bind to Robo1, that block the DCC, so natrium cannot bind to DCC. If natrium cannot bind to DCC, it will move straight because natrium is not going to show any attractive effect, and the SMO is not allowing it to move on that side. Making sense? So it will keep on moving down. When the time comes to move cross the midline, at that time, Robo3 start expressing, and Robo3 activity does two things. Number one, it goes to send signal to nucleus, and nucleus start making protein 1433. 1433 come out of the nucleus, goes to SMO and block the SMO. So the natrium chemoattraction is blocked. The other thing it does is, it Robo3 activation block the Robo1. If the Robo1 is blocked, that means the SMO cannot, slit cannot bind here. If slit cannot bind here, Robo1 is not active. If Robo1 is not active, then the blockade on DCC is gone. So natrium from here can come and bind to DCC. Now you are getting an attractive signal toward the midline. So now it moves toward the midline. Now when it is at the midline, you want to move it away, further away from the midline. So what that happened, during that time, Robo3 signaling goes down. When Robo3 is going down, that means this slit, this Robo1 will be, again that blockade will remove, slit will bind to Robo1, it's binding there, it's say go away from here. And when Robo1 is active, that blocking DCC, so when DCC is active, blocked, natrium cannot bind it, so there is no attractive signal left here. Now, Robo3 was down, 1433 is also gone, so here sonic hedgehog is causing it attracting, and slit is telling it to go away, so it cannot actually turn back. So it has only way to move on this side. Making sense? Any question? Okay, talk to each other for good 2-3 minutes so that these steps are clear.
It's just the gradient.
That's what attracts it to go down.
And then, when it wants to start moving in, the axon starts to press it. Blocks it over the line, and activate. Then there will be.
Well, we have to download this picture.
We mean one mil and then small.
So
I mean,
either
Are you like translating? Yeah.
I really like Berkeley.
When are you, will you be here in May?
Oh, we say that.
I mean, I'm on track, so I think we're just gonna talk about what else we should add on to do. Like, just making sure.
Yeah.
Yeah, that's why I don't really want Tuesday.
I don't wanna wait till like Tuesday, cause I'm not in Boston. Cause like, I'm not having allergies to spend it.
So then, three blocks for the one, and then you also have 33 blocks as Thursday. Now, ECC is open for
So just the
entrance.
same, and all the rules as well.
Yeah, because all the railroads are separate. Railroads are separate, just like,
Yeah, the same thing.
Yeah.
So then the sign that you
it just depends on the expression.
And
Well,
that's why
it's-
we get active
it's there,
too.
and it's small, it's activated, but then
Oh, just the 14, okay.
Negative.
And then, go to 50,000.
Any doubts, please? You okay? Any doubt? So we are saying that these axons are moving, right? So how these axons they travel? We said there are signals which are telling them attractive, or signal they are telling them repellent, right? But if you think about this whole, just think this is the whole embryo or a limboid, right? So the axons, they're not one axon, there are many neurons. For example, there are 10,000 motor neurons there, right? So 10,000 motor neuron is going to send an axon like this. But these axons, they cannot travel everywhere, okay? So the axon, so we are talking about cell adhesion is a mechanism to travel. Essentially, that's what I want to talk about. Okay? And to travel means how they find the road, right? How they find the road. What is road? On which they need to travel. So what happened that these axons, for example, if this is your growth cone, and these axons, they actually show some proteins on the cell surface. So right here, you will have some integrins, and you will have neuronal N-cadherins. Now these proteins will allow them to stick to the particular proteins which are on which they can travel. So axons travel on sticky surface. Therefore, growth cone can grab the road. Now, so what is that sticky surface? The sticky surface is neuronal laminin. So, if this organ is there, which we just drawing, they are not going to travel like this. They are going to travel only on the area, for example, this area, only this area. This area has laminin. So they will actually, so what is going to happen? Let me draw it here. So if this is a growth cone coming there, this growth cone is coming there, this is growth cone, and it coming here, and it says, okay, this area doesn't have any laminin. So it touched there, this area doesn't have any laminin. So what is going to happen? It is going to retract. So that's what we are showing in the movie, like they're forming, they will retract, they're going further, right? So it will retract back. But if we find an area, for example, here the laminin is there, it is going to grab that, okay, and move on that track only. So it will not divert on this side, it will not divert on this side or this side, it will stay on the laminin track. So if it goes like this, it's going to retract back, okay? Now, these axons which are there, they don't travel like separate axon. They travel, so this axon is there, this axon is there, they don't travel like this. What they do, they travel like in the bundles. They will be in the bundle. So this process where you have separate axon like this, and they basically form bundle, this is called fasciculation. Now, each of this axon can have different target. Some might have this muscle target, some might have this muscle target, some might have this muscle target. But in this fasciculated axon, this is actually called a nerve. So nerve is a bundle of axons, or it can be dendrites depending on what type of neuron you're talking about. Okay? So bundle of axon or dendrite is a nerve. So all those nerves are there, but the target of each axon is separate. So for example, this is the target A. This is target B. So what is going to happen when this nerve is coming like this, right here is the target, and it's actually going to go, one axon is which is the target from that neuron going to go here. It's saying this is my target, this is going to go like this. So this process is called defasciculation. Defasciculation. And this is done by polysialic acid. Okay? And this process is regulated by BMPs. So if you want to read more about this, we did a paper a long time ago on this, how this thing happened. If you go to PubMed, you put my last name and just do BMP. So go to PubMed, write my last name and BMP, you will find the whole paper where we have discovered this whole phenomenon, how that happens. Okay? So that's why I teach developmental biology, because I've worked a lot on this. So this is how they actually travel along. Okay? Now, so we said there are three things we said. Question?
No, it's okay.
Okay. So in the beginning we said they should know what the target is. There should be some attraction, right? We said trophic factors are there. Then how to go to target, you should have some lights and all those things there, which are chemoattractant and chemorepellent. Now what are roads? Roads are the laminin, and you should have bundling and defasciculation to reach the target. Now when they reach the target, how the neuron talk to the target? That's what we want to talk. What is that connection? How to find the right address? Okay? So you can talk about this for a minute if there is any doubt. Yeah.
Can you re-talk about the defasciculation?
So defasciculation is, so fasciculation is bundling. Defasciculation is, for example, it's going there, this is a bundle, and say this is my target, right? So there are maybe 20 axons there together. And for this neuron, this is the target. So what is happening? So this come out of the bundle and say, okay, go to target. That is defasciculation. So bundling is fasciculation. You lose the bundling, that is defasciculation.
And what, like,
And
what is?
you might have seen in your culture also, when you're taking the picture, you might have seen 20 neuron group and you will see one common thread coming out. So each of these is having axon, but those axons have fasciculated. All right, so what was the next?
So what is like, so the
Polycylic acid causes defasciculation.
okay, was there another thing that was?
BMP is regulated.
BMP is,
Yeah, the BMP, we know about BMP proteins, right? So BMP, Nogin signaling, that actually regulates this. So let me just give you a very simple example. I want to draw a face. Okay. So this is the face, okay, these are some eyes there. Okay. So a common nerve come here, a big nerve come here, right? I'm forgetting submaxillary or something, I'm forgetting the name of the nerve. Now that single nerve is coming there. That nerve goes to eye, cause optic nerve. That goes to your facial bones, maxillary nerve. And then these are your jaws. These are your teeth. And the same nerve from here fasciculating here. So one nerve bundle coming and fasciculating different area. And here also from here going to upper giant lobula. This is all fasciculation, defasciculation. So one single nerve give to 3 or 4. All the facial fingers essentially controlled by one nerve that divide into different areas. So I should not say divide, actually defasciculate in different areas. Okay? Okay, now they have reached the target, so they have to connect the target. So when they reach the target, they form synapse, and I'm going to talk about synapse formation. So when they are reaching, so synapses, when an axon contact target, and that target can be a muscle or another neuron, anything. Okay? So they form a specialized structure. That specialized structure is called synapse. So when it formed. So how the synapse is formed? So think about, for example, say, so I'm trying to draw a three-dimensional thing like this. Say this is the muscle fiber. So this is one muscle fiber. And to this muscle fiber, at that time when muscle is also being divided, also being developed, so this will not be called muscle fiber at that time. So at that time we'll call it a myotube. I should write developing muscle fiber called myotube. So they are also developing. And this developing myotube, so on the muscle, when the neuron go to muscle, all these neurons are cholinergic. Cholinergic means they are making acetylcholine. Okay? So, for example, let me draw it here. Say this is the axon, and that axon, this is the growth hormone there, right? So this is the axon, and this is the growth hormone. And all those neurons which are impinging on the skeletal muscle, those are cholinergic neurons. And cholinergic neurons means they make acetylcholine. Now acetylcholine is a neurotransmitter, right? Acetylcholine neurotransmitter. And acetylcholine neurotransmitter is in the vesicle. So these vesicles are there. So I'm writing here neurotransmitter vesicles. So neurotransmitter vesicles are. Okay. Now, so if the neurotransmitter vesicles are there, it's the acetylcholine, then on the muscle you should have the receptors for the acetylcholine, right? So on the muscle, right here, so these are acetylcholine receptors. Okay. Now what happens? So it is coming there. Acetylcholine receptors are kind of spread out. Okay. So what happened that the nerve terminal, which the axon terminal, I will say, at that time, because it has reached here. So the axon terminal secretes a protein called agrin. So it secretes a protein called agrin. Now agrin is secreted here, so this is agrin. So agrin is secreted there. Now, what happened when the agrin is secreted there? What agrin does, as the name suggests, agrin, it aggregates. What it does, all these acetylcholine receptors, which were like far away spread out, they aggregate under the nerve terminal. So what is going to happen here? So all of these acetylcholine receptors that cause aggregation of AChR, acetylcholine receptor aggregation takes place. Okay? And then what happened? These neuroterminal vesicles, which were here in the axon, the neurotransmitter vesicle. So neurotransmitter vesicle move to the axon terminal. Okay. Now, on the axon, remember there were two types of protein we talked about. Right here. There is N-cadherin there, and there is fibronectin also there. And on the, again, where they will go? They will go where the laminin is there, right? So the muscle, right here on the muscle, will have neuron specific laminin. Muscle will have neuron specific laminin. So what is going to happen? That is going to cause this. So now what is going to happen? You have your muscle there, and that is going to cause the attachment of this axon terminal with the muscle, right? And then what is going to happen? This whole structure will spread. So this whole structure will spread. Neurotransmitters are there, neurotransmitter vesicle is there, and now there is that connection being made and spread, and axon terminal will spread. Okay? And then what happened? Then each of this place where this thing is, that this membrane kind of convolute like this. Let me draw it here. So say it's like this. It convolute like this. And in this, each of this area, now I'm kind of like shrinking it like this. Okay? So each of this area, so now you have one axon coming there, and from this axon, this branch kind of like go like this branch go like this, and kind of this branch go like this. So this kind of division takes place. Now what happened? After this, the cells called another cell come here. These cells are called Schwann cell, SCHWA. Schwann cells come and they encircle the whole area, and ultimately this whole area is covered. And Schwann cells are also responsible spreading the myelin sheath around those. Now, what can happen? You can have, I'm just trying, you can have this muscle fiber, this is another muscle fiber, this is another muscle fiber, this is another muscle fiber, multiple muscle fibers. Now, here is one axon, here is another axon, here is another axon, here is another axon. So this axon can make a branch, can make connection here, make a connection here. This can also make a connection here, make a connection here, or make a connection here, make a connection here, make a connection here, make a connection. Multiple connection it can do in this stage. That is what is happening in the development. So you can have multiple neuron, this is important thing, multiple neuron making connections on the single muscle fiber. That can happen during the development. But only one axon can make a connection to one muscle fiber. Making sense? In the development, multiple axon, for example, this axon is also making connection here, this axon is also making connection to here, this, all are making connection to this one fiber in the development. But in reality, there can be only one axon connection per muscle fiber. This is one axon, right? This axon divided, making three connections, but still one axon on one muscle fiber. This is the real situation which should happen. But they make many connections here. Then what happens? Have you seen the little baby? You go to baby and you smile at the baby and you say, "Shh, baby smile." Right? And you feel very happy. Oh wow, baby is smiling, making you happy. Then baby is moving legs, arms like this, like crazy, right? And you say baby is very happy, baby is moving arms, legs, and all those things. That is the process at that time. The developing embryo is already developed. It is refining the connection. So most of the connections, it's kind of firing. So that is especially for the skeletal muscle. It's firing at that time. So at that time, all but one connection will remain. So there were studies in the early times. Now we have ethical issues. What they did, they basically tied the baby. So baby could not move the legs and all those things. Those babies were severely mentally retarded. So each of these connections, think about if you're moving this, you are thinking in your brain you want to move it. Brain is sending signal to spinal cord is sending signal here, it's moving. When it's moving, it is touching so many things, you have sensory signal. So all that circuitry of the nervous system is actually tested when baby is doing these things. And if you don't let baby to do those things, that circuitry is messed up. And that's why you have lot of things. You have, attention deficits in kids, and so many type of diseases are there, right? Those are all the synaptic problems. Most of those diseases are synaptic problems. If you don't have the right connection. So that's why, you remember, it's not happening now, but if you talk to your grandparents, they will say in the old time they used to have like so many things around the baby. So people used to give even the books to, I don't know, in say 10 years ago, when the baby is born in the hospital, they used to give baby book, even when the baby could not see the color. They'll just say, show baby every time the different colored book and all those things. Because all those are kind of helping baby to refine the connections. So have a lot of things in baby's room, a lot of colorful books, lot of things so that the brain can be activated. And that's why they say it's good to talk to other people because they will have more company. So avoid cell phones, avoid all those things. So interaction is more important than these things. Okay? So with this, we are done with the development of nervous system. Okay. So this topic is done, and we will talk about how much time we have. We have time, so.
Like 5 minutes.
Oh, so we can. I'll just give you something. So just talk about this part.
He said it doesn't work. Yeah.
So he was saying like when it's in development, or so
Or
in
like
the
you said,
end,
they can hide it,
like if you tie the baby down,
Yeah.
it doesn't lose any of its bones, like the connections on like the baby doesn't understand how.
And then once that happens, it's just these grooves going through the axon, basically splits, and then Schwann cells.
Yeah, Schwann cell covers the whole structure as well as the axon, because Schwann cells are going to give you the
I
myelin
don't
sheath.
know how you guys take notes on pencil, like I just copy and
And
paste.
myelin sheath is important for the nerve, for the fast nerve
Yeah, I
conduction.
did
So
that. This one was there.
just, so you can just imagine how fast nerve conduction is 100 meters per second.
I can't do.
Huh?
So
I can't do the left side.
you can think like someone touching and you immediately respond. So how
Whenever
fast
I'm
is
writing
she?
on paper now and I make a mistake,
So
I
that's
just go
because of
like
the
that.
myelin sheath, which is
And
secreted
I'm
by
like,
the
wait a
Schwann
second, I
cell.
have to erase.
So the multiple sclerosis is a disease. Have you heard of that disease?
I mean, like the
So that is
eyelids,
because
like,
the
sucked up
demyelination
or
process,
so, like,
myelin
straight.
sheath is not formed.
But he got like a line.
Or it's not formed properly.
You know that
Or
you can get like a cover.
there are so many, like
yeah,
provide
I
is
have that
one of
feels
the
like
things, and
paper.
our food does so many bad things.
I
So
can
provide
probably-
is there in
Cause
the
that's
food,
what
that
Megan,
provide can somehow
Megan has like a
cause
cover that feels
And
like paper.
so Schwann
so
cell
that
can
when she
divide
writes
and
on it,
make
it feels
more,
like
that's
turning
why
paper.
if you heard
I
of
had two.
patients
And it was
story,
nice,
multiple
but
sclerosis, they
it
will
got
say, they will
peeling
find
'cause
this,
this is like
fine
right
for
at the
6
edge,
months, then
it's
come
kinda
back.
Magic.
Then again, fine for 6 months, then come back. So the
I don't
Schwann cell
know.
is not quite right.
Yeah, I can't do that.
And
No,
the formation of these
very
like grooves
bad.
in the muscle fiber was
I tried to exchange my iPads, like,
sections.
I don't know, before this year started, I was like.
Well, no,
I'm already
we are
there.
saying like once this
You
is
know, like,
spread out,
it's for sure.
Yeah.
once that's spread
I
out,
just, like,
that lead to the
exchange it,
formation
and if I
of
really do believe
these
in all
groups
that
in the
shit,
network,
we would have
and
like
it
an
goes
iPad.
basically
Yeah.
sit.
And they're like, no, it's still school property. I was like, yeah.
And there are,
But I'm
I did talk about, so biofilm
Yeah,
and
and
all
you
those
also
things, they put that
cut off
in.
your
Also, yeah.
Yeah.
Which I feel like, I feel like it was archived.
Question here?
Oh, where do the Schwann cells come
Hmm?
from? Because isn't the axon like extending? Where do the Schwann cells come from?
Schwann cell are developing, so, oh yeah, we didn't talk about, that's what we wanted to do in the lab, but we never reached there. So, neural stem cell, let me just show you for a minute. So this stem cell, for example, divide in 2 areas. Say it divide like this, then you have multiple cells formed, right? Like this. So then these are expressing notch there. So this cell is, for example, expressing notch, right? And the other cell right here on it, that will be expressing the receptor for notch. It can be jagged, it can be serrate. There's one more, I'm forgetting the name. There are 3 receptors for that, for the notch. So what happened when this bind to that? So these are stem cells, right? So what happened, this notch cleavage take place, this notch is cleaved. And then this notch goes to the nucleus. Now, this decides that which cell is going to become neuron, which is going to become the glia. So this cell is going there inside the nucleus, making all those things, hey, saying you become neuron. So notch signaling is activated. And the cell which is not having notch signaling, you become glia. So side by side, you have glia and notch, glia and neuron produced. So they are produced in the same way. And what happened when the axon is growing? Now glia, initially people used to think that glia are just supplying the energy, trophic factor, sport. It's true, but they have way more than that functions. So these cells, they migrate along as the axon is migrating out. And you can have this thing, and you have this axon, and you can have a Schwann cell right here, surrounding it. Another Schwann cell surrounding it. Another Schwann cell surrounding it like this. And these Schwann cells, they secrete myelin. So you will have myelin there. And then Schwann cell at the tip also, and they basically encircle it. So Schwann cell are developing along with the neuron. So that's why in that neurosphere, you might see 10,000 cells, but you see other cell also, but we are seeing only some neurons, right? So other cell might have become astrocyte, other glial cell, though we didn't stream for those in your culture system. Remember, if you take those, you will see other cells also there. But our antibody is staining only for neuron. We didn't stain for the glial cell. So they would have been other glial cells. That is making sense. Any other doubt or any other question? Okay, so we'll talk about so we're done with this. In the next lectures, I don't know how many we have left. We have 2 before Thanksgiving, I believe so.
Yeah.
2 before Thanksgiving. And after Thanksgiving, we have 2 or 3 weeks probably. 2 weeks. So we have 6 lectures after. So in those 8 lectures, we are going to talk about all mesoderm, like how the muscles are formed, how the bone is formed, how the vertebrae is formed, and how the kidney is formed. So we'll try to cover at least these 4 things.
So will we have like another test after Thanksgiving?
Yes, we'll have another text after Thanksgiving of this topic only.
And then
Just
our-
this
Only
this
this
topic.
topic?
Only this topic, means whatever we have covered after the exam period. I think we covered the neuron alone.
So are we gonna have?
So like Friday and Monday's lecture, are they gonna be all?
Friday lecture will not cover the test, right? Friday
So
will
just
start
this
the new
topic.
thing.
So it would just be one topic?
Yeah, because the new topic will not, that will be our last topic, we will finish in the end,
Okay.
Okay.
right?
So our test is
After Thanksgiving?
After Thanksgiving.
Okay.
like just.
Or you want to be tested on Monday? I don't
I
want.
won't be here Monday.
I don't.
So let's start the new topic, and then we will test it after on that topic, on the neuron.
Okay.